All right, this is chapter 25, part 2. We left off here on phylograms. So phylograms are a specific type of cladogram that looks at changes in DNA or RNA sequences. So here, for example, we have Drosophila, lancelet fish, and the length of the lines refers to the amount of change or the amount of differences genetically in one specific sequence or one specific gene. So if you look at, for example, amphibian and mouse at the same gene that is conserved in both, the amphibian, from its common ancestral gene, the amphibian has had more changes than the mouse, and thus the amphibian has changed faster. That genetic sequence has changed faster than the mouse. This is a phylogram. An ultrametric tree is the same as a phylogram, but the length of lines don't mean anything. They're all the same equal length. It refers largely to time periods here, as you can see, Paleozoic, Mesozoic, and so on. Now, usually there's some debate as to the proper structure with a cladogram, even between evolutionary biologists and so on. So we have to take whatever data or information we have and apply it as best we can to create this cladogram, to look at these relationships. So we apply these things called maximum likelihood and maximum parsimony. The most parsimonious tree is the one that requires the fewest evolutionary events to have occurred in the form of shared derived characteristics or in the form of differences. Now, parsimonious means the simplest or most logical. So the simplest or most logical is the one that requires the least number of events or changes to have occurred for it to exist. So for example, here we have some data. We won't get into the data, but you can take a look at that specifically. Here we see the difference between human and mushroom is 30% percentage of differences. And using that data, we can compare or create two cladograms. One here, okay, tree 1, and tree 2 is also correct, although very different, as you can tell. Here we show humans and mushrooms sharing a more recent common ancestor, and here we show humans and tulips sharing a more recent common ancestor. So we're going to choose the one that's most parsimonious or most likely here, which being tree 1. So maximum likelihood basically is using the certain rules with DNA changes over time. You use the one that reflects the most likely sequence of evolutionary events. Here we see species 1 through 4 of these different birds, and using the data, there's all different kinds of possibilities where you notice a comparison of number of changes, DNA changes, to create these different branch points. And based on all these numbers of events, number of changes that must occur, we find that this tree is the most likely, or the greatest likelihood. You can see based on the orange lines here, and compare that to these, you notice this is the most likely, the fewest number of changes, essentially. And so all these things are just hypotheses, right? You use morphological, molecular, and even fossil evidence when you have it to create the best. Now, of course, not always is the most parsimonious the best hypothesis. Okay, here you compare mammal and bird clade, where mammals are more share a more recent common ancestor due to they both having a 4-chambered heart, whereas lizards do not. Both birds and mammals have a 4-chambered heart. Versus here, lizard-bird clade, where you have lizards and birds being more similar than birds and mammals, even though they both share a 4-chambered heart. There's other characteristics that make them more likely to be similar. So typically we use a lot of molecular evidence now to look at relatedness. Now there's a lot of genetic material available for us, in part due to gene duplication, where as additional genes are added into the genome, there's more locations or spots for mutations to occur, and thus to notice or to see, or for evolutionary changes to occur. So here we have one example, one type of duplication, and these are called orthologous genes. Now, orthologous genes are genes found as a single copy in the genome. Okay, they're only going to diverge when speciation has taken place. So here's the ancestral gene. Here's 2 orthologous genes, and you notice within that there's these 2 genes specifically. And then you have paralogous genes, on the other hand, which also results from gene duplication. But they're found in more than one copy in the genome. And they'll diverge within the clade that specifically carries them. And usually they are associated or enable new functions to occur. Here's your one ancestral gene, and different regions. Specifically here you see 3 and here's 2. So orthologous genes are really common across all different types of species. And these species can be varying in complexity. And all it basically says is that the genes in these complex organisms are very versatile, and that one gene can perform many functions. So looking at genome evolution. Now, using this molecular data, we can create a molecular clock or yardstick where we can measure the absolute time based on numbers of changes or mutations. Now, we know that mutations occur on average at a sort of a predefined rate. We know, for example, that DNA polymerase III has mutations or adds a mistake, an incorrect nucleotide, causes a mutation every 1 × 10⁹ duplications or nucleotide insertions. So we can basically infer this constant rate and create a molecular clock for these changes. And so then based on that, there's something called neutral theory, which essentially says that evolutionary change in genes, and of course proteins, they don't have any effect on fitness, and thus natural selection doesn't play a role. And thus the rate of molecular change in these genes should be just like a clock. So the molecular clock is sort of based on neutral theory. Now, we of course know that this is not the case, because we know that changes in genes does affect fitness and is affected by natural selection or play a role in natural selection. So the molecular clock is not really as smooth as neutral theory would like to predict. However, even so, we can take this example here where we take the origin of HIV and look at its molecular clock, where HIV is descended from viruses that infected chimps and other primates. And in fact, going even further back, for example, there's cat FIV. It's called FIV, feline immunodeficiency virus. And so what they do here is they take HIV samples throughout, and they compare the sequence of those samples and look at the number of changes. And it shows that it's pretty clock-like in their number of changes here, where you can look at, here we have the different domains, bacteria, archaea, and eukarya. And this is essentially a tree of life, and showing where these different branch points would occur. If you were to look more specifically at this bacterial sequence, you could create a even more defined molecular clock. But here, this is just showing a generic molecular clock, if you like. Number of years here is present day. Typically, the end of these lines are present day, where this is nearly 4 billion years ago, the origin of life. And we're done.